So we left off here, we were part the way through this first packet, which really was going through the functional anatomy. So the major functions, the major structures rather of the GI system, the GI tract, and then some functions that are associated with each of those parts. And as I was mentioning before, this is really a review. So I'm not going to spend a lot of time going into too much detail here because this is a lot of what we talked about in the GI system lab. So we left off here at this slide looking at the large intestines. And we know the parts of the large intestine. We've got the cecum, which is the first part of the ascending colon. This is where we find the appendix. We've got the colon, which is the remainder of that structure, which is divided into the ascending colon, the transverse colon, and then the descending colon. We also want to point out the ileocecal sphincter. So the connection between the cecum and the ileum being the terminal part of the small intestine, there is a sphincter, a strip of smooth muscle located there. And so the emptying of the small intestine into the large intestine is regulated. Okay, so depending on different emptying times, depending on the density of the food, that is allowing enough time for absorption to take place before the contents of the ileum are dumped into the cecum to get into the colon. And that's the purpose of that ileocecal sphincter. We also have Taenia coli being a unique feature in the large intestine. These are those bands or strips of smooth muscle that run longitudinally throughout the large intestine, and this is what gives the large intestine this kind of gathered appearance, which we call haustra. And so the colon, which is that entire length of the intestine, is really responsible for concentrating the waste, solidification of the waste, and absorbing most of the water that is present in the chyme, right, the mixture at this point, so that it can be solidified to be eliminated via defecation, okay? So the large intestine, really important for water reabsorption, which is a part of conservation of water. So that's how we also regulate our water balance, reabsorption of most of that water in the distal part of the intestine, as well as solidification of that, that content to become waste. Okay, so we talked about these four segments, the sigmoid colon being the distal part of the descending colon, which is then converted into the rectum. And then finally the anus, which is the distalmost ending of the large intestine. So let's talk a little bit here about the rectum and anus. The colon empties into the rectum, right? We talked about feces being solidified and excreted through the anus. These are the parts that make up the large part of the intestine. We have two important sphincters that are located here. There's an internal anal sphincter, which is again, a strip of smooth muscle that wraps around this part of the rectum. And then there's an external anal sphincter, which is skeletal muscle. So the internal sphincter is under autonomic or involuntary control, and then the external sphincter is under our voluntary control because it is made of smooth muscle. So this is where you can consciously decide to relieve yourself via the control that we have over this external sphincter. So in order for defecation to be complete, we've got to relax both the internal sphincter as well as the external sphincter. So there is an autonomic component to the defecation process, right? This has to come through autonomic fibers, which are coming from the integrating centers in the brain, depending on certain stretch receptors that are located in the colon. Okay. We're going to spend the last part of this packet speaking about some of the accessory glands. So we've got four main glands here that are part of the GI system. We've got the salivary glands that release their secretions in the oral cavity. We've got the pancreas that releases pancreatic digestive juices into the second part of the intestine, so the duodenum. Excuse me, the first part of the intestine, my bad, the duodenum. We've also got acini. So acini are basically these glandular structures that make up the liver that help to produce bile. And so the acini are a part of the liver structures where that bile is made and then secreted and stored in the gallbladder. So this is what a gland looks like. These are the acini that we're speaking about. So these are the actual cells. These are epithelial cells that are lining these ducts. And so these are the acini cells that are actually making the secretions that are being released. They get released into these ducts, and then they make their way into the lumen of the GI tract. So the body of the gland, any gland, is really just a series of acini cells that are making these secretory products that then make their way via these ducts, glandular ducts, into the surface that they are excreting those secretions onto. Okay, and then along the ducts, we have modification of those secretions by transport mechanisms. So we can make those secretions more watery, more viscous, depending on where the specific secretion is. An example, saliva is made to be more watery so that it can flow through the duct and make its way into the oral cavity. In parts of the intestines and stomach, the secretions are made to be more viscous so they can protect against the acid. So there are modifications that are made based upon different transport mechanisms that can make the primary secretions from the acini cells either more viscous or more watery, depending on where that secretion is and what's needed. All right, let's see, I have a question here. Oh, okay. Okay, so someone was sharing some more feedback there, all right. Okay, let's keep it moving here. So let's speak about some of the characteristics of saliva. So first of all, it's rich in bicarbonate. It also contains quite a bit of mucus, as well as some of the enzymes that are necessary for digesting starch, so salivary amylase, but also a part of the immune system. So lysozymes are protective enzymes that help to degrade bacteria, viruses, pathogens that may be entering the oral cavity with food that we're ingesting. So the enzymes are not only breaking down food, starch specifically, but also protecting from pathogens that may be in that food. Let's talk about the pancreas. So the pancreas is another one of those accessory glands. The pancreas has both exocrine and endocrine function. When we looked at the endocrine system last semester, we talked about the islets of Langerhans, which are the clusters of endocrine cells, alpha, beta cells that secrete insulin that are found throughout the pancreas. That is not to be confused with the exocrine part of the pancreas that we're talking about now. So the exocrine part of the pancreas uses ducts for its secretion, and those secretions are pretty much bicarbonate, which is helping to neutralize the acidity of the chyme that's coming in from the stomach. There's also other substances such as amylase, breaking down starch further, lipases, which are breaking down fats, proteases, breaking down proteins, nucleases, breaking down nucleic acids. And so the pancreas is one of these organs that generates quite a bit of these digestive enzymes that break down a lot of the components of our food. Fats, proteins, you name it, right? Starch are all broken down with the digestive secretions from the pancreas. So relative to its weight, the pancreas secretes more protein than any other tissue in the body. So it does a large job in breaking down most of the proteins that we eat. Different forms of protein, so plant protein, animal protein are broken down in different ways because of all these enzymes that are present. So here what we can see in this schematic diagram, we see the pancreas, which is this organ here. It has the head tightly or intimately connected to the second part of the duodenum, or the first part of the intestine. So this is the duodenum here. And then we have this duct that is releasing these secretions into the duodenum. And then these secretions are pretty much made up of these substances, bicarbonate and all of these enzymes that we mentioned. We can also see the common bile duct being opened into the duodenum here as well. So this is a shared opening between the pancreatic duct and the common bile duct, which are both releasing their secretions into the duodenum. If we zoom in a little bit closer at the pancreas, we can see the acinar cells that I mentioned earlier. These are the actual cells that are secreting or producing these enzymes. And then the enzymes are dumped into the first part of this, this duct. And then as these substances make their way to the lumen of the GI, they are being modified by secreting either bicarbonate or by making these a little bit more liquidy so that they can flow appropriately towards their opening into the duodenum. The liver is another accessory gland. A lot of people don't know this, but the liver is a gland. It's a large gland. So most of the liver tissue is pretty much just these acinized cells that I talked about, as well as the ducts that are draining those secretions towards the various openings into the common bile duct, etc. So it secretes bile as one of its main functions. It also is important in processing nutrients. So as I mentioned before, we left off speaking about this on Monday, the food that we eat is broken down to smaller parts, absorbed across the GI epithelia, mobilized into the blood that is in the venous system, and then that blood is taken to the liver to be detoxified and to be processed. And so the liver is going to take the glucose that's absorbed and make it into glycogen, store it into glycogen. Most of the glycogen storage happens in the liver. It's going to take those amino acids and they can be converted into fatty acids depending on the body's needs and the body's metabolic demands at that time. We can take those triglycerides and cholesterol and convert them either into their storage form or into lipoproteins that can easily be circulated through the body. So basically the liver is taking all of these micromolecules, processing them appropriately according to our metabolic state, and then either storing them or circulating them to other parts of the body.